Neurotransmitters and medicine. In this lecture, we'll be discussing how medicines affect the synapse. By the end of this lecture, you should be able to define neurotransmitter, excitation, inhibition, acetylcholinesterase, SSRI, MAOI, and opioid. Be able to list the events at a synapse. Be able to list major neurotransmitters. Differentiate between types of psychoactive medications and drugs, SSRIs, MAOIs, opioids, and alcohol, and explain the causes of depression and anxiety from a neuronal perspective. You may notice that there are several objectives here that overlap with a previous lecture topic. That's because the first thing we're going to do is review the events of the synapse in class. You, however, online student, have access to the other lecture and can just review it by watching that or by coming and talking to your professor. So we move down to here. And what happens to neurotransmitters? Well, they are released into the synaptic cleft, and there they will bind to a receptor on the neuron on the other side of the synaptic cleft. And after that, they probably won't diffuse away. Usually they will be degraded, or they'll be taken back up by the neuron that released them. This will be important as we look at different neurotransmitters and different drugs. So, what does medicine do at a synapse? First thing it can do is prevent the release of neurotransmitters. We're not going to really cover those today. It can, however, act on neurotransmitter actions within the gap and do something that would happen to a neurotransmitter. And it can, of course, bind to receptors on the other neuron. We're going to learn the different neurotransmitters and the drugs that are similar and affect them. So we're going to look through acetylcholine, the amino acids GABA and glutamate, and then the biogenic amines, norepinephrine and epinephrine, dopamine and serotonin. We're not looking at neuropeptides or gases in this lecture. First off, acetylcholine. You may remember acetylcholine from the neuromuscular junction, where a neuron will innervate a muscle and cause the muscle to contract. Acetylcholine is released at the end of the axon and will actually bind to a muscle. This causes a depolarization of skeletal muscle cells and initiates contraction. It is also present in the brain for memory formation and learning. That's right, it's in your brain right now. This can also slow heart cell contractions. Now, acetylcholine is digested by an enzyme called acetylcholinesterase. This will break down the acetylcholine in the synapse. And what insecticides can do is actually overload the nervous system by affecting the degradation of acetylcholine. Now, faster in insects than in humans, of course. They will cause blurred vision, headaches, and rapid pulse and sweating in humans when there's too much exposure. In insects, they just kind of paralyze them. There are also several other things that we will be discussing in class that can affect the acetylcholine at the neuromuscular junction, but we're not really going to look over those for too much import. Next up are epinephrine and norepinephrine. So these mediate the fight or flight response. You may be familiar with adrenaline, and they do this through multiple different mechanisms. We can see here this example we had back from the receptors lecture, where epinephrine can bind to a beta receptor in either liver cells or the smooth muscle cells in the wall of blood vessels. So in either way, you have different actions. In the liver cell, blood glucose level will increase as a result of epinephrine binding to the beta receptor, and in the smooth muscle cells of the walls of blood vessels supplying skeletal muscles, it will cause dilation when epinephrine binds to a beta receptor. However, we have epinephrine binding to an alpha receptor in the wall of blood vessels that supply the intestines, and that causes a constriction, decreasing the flow and stopping the rest and digest. The difference between epinephrine and norepinephrine is simply a little methyl group, which is this little arrow here attached to the N. They do similar things. Next up are our amino acids, glutamate and GABA. This is what glutamate looks like, and you see that it has the amino group here, and it has an acid group, and that makes it an amino acid type of neurotransmitter. We also have gamma amino butyric acid, which is GABA, or GABA for short. These are actually affected by ethanol because ethanol looks very much to your brain like GABA. There's GABA with that OH. There's ethanol with an OH. Yes, it doesn't look too similar to you, but to your brain it's close enough. And it binds to the GABA receptors in the brain specifically and inhibits normal GABA functioning. This will also inhibit glutamate receptors. And what happens here is you get drowsy, you lose inhibitions, and all the other things that happen when we drink alcohol. So this would be a good moment talking to college students to discuss how much is too much and to provide you with a nice chart. Notice at 0 drinks, both women and the men are perfectly 0% blood alcohol. As your weight increases, the amount of blood alcohol in your body by a single drink decreases. So this means that people who weigh more will respond slower, generally and not always. You probably do know of at least one person who's very small and seems to be able to pound them away all night, or someone who's very large and seems very intoxicated very quickly. That's why this would be a good moment to also say, hey, look at this chart. It's not always true for everyone, and it says nothing for what a hangover does. What it does show is that in a couple drinks in an hour, you're going to be increasing your blood alcohol until you are not able to drive, and it doesn't take a lot of drinks for that. Next up, serotonin. This is made from tryptophan, an amino acid. It's related to sleep and related to mood. Levels are reduced by insufficient sleep, sugar instability, i.e. being hangry, and chronic stress. Well, we also know the insufficient sleep, sugar instability, and chronic stress as college. So serotonin is actually affects mood, and it is affected by selective serotonin reuptake inhibitors. When serotonin is released in the synapse, it is then taken back up by these uptake proteins, and SSRIs such as Zoloft, Lexapro, and Prozac will inhibit the ability of these proteins to take serotonin back up. That means that serotonin will remain in the synaptic cleft for a longer period of time. It will bind to the receptor, jump off, and then bind back, as opposed to bind to the receptor, jump off, and be taken back up. This will cause, of course, better sleep, better mood. So this is why this is an antidepressant. Dopamine is made from tyrosine, which is also an amino acid. This is known as the pleasure molecule. It is involved in motivation. People get dopamine, well, a lot of animals get dopamine when they do something good. You're doing a good job by watching this lecture and by making sure you're taking notes. You're doing a very good job. See, I just released a little bit of dopamine in your brain. Too little dopamine causes depression. Too much dopamine actually causes schizophrenia. And dopamine reward pathways are hijacked during addiction. Dopamine can be affected in the synaptic cleft by monoamine oxidase inhibitors, or MAOIs, such as Marplan, Nardil, and MSAM. These decrease the breakdown of dopamine, causing higher levels to stay in the cleft, because normally dopamine is broken down by monoamine oxidases. Mescaline is a specific drug that stimulates the dopamine and the serotonin receptors by binding to a specific receptor in the brain, your 5-HT2A-C receptor. So mescaline binds to this and affects the serotonin and dopamine levels. Now you might think monoamine oxidase inhibitors, well, that'll stop the breakdown, and selective serotonin reuptake inhibitors, that'll stop the reuptake. What if I take both at once? Short answer, don't. Anyway, next, opioids. For opioids, these are going to decrease your body's awareness of painful stimulation. This doesn't decrease the pain, just one's awareness of it. It will also decrease respiration, which can get dangerous at high levels, and it increases dopamine release, causing a sleepy and relaxing high. The way it does that is it's going to actually stimulate the GABA receptors to cause a release of dopamine in these nerve terminals here. Now, when an individual stops taking opioids, there tends to be a lower level of pleasure because the body has adapted to a higher level of constitutive dopamine. So this also causes pain all over because the individual no longer has blocked pain receptors. They overcompensate by feeling pain all over. There are gastrointestinal upsets at extreme levels, and we can discuss that a little more in class because that's an interaction between two different systems. Goosebumps like a plucked turkey, which is why the term cold turkey is used, and a general feeling of restlessness. I will be discussing what this little picture means. It's from a book series I love, but I'll be discussing that in class. Let's look at these different drugs here, and let's look at different toxicities. What you can see is that heroin causes a great deal of physical harm and is very easily causing dependence. It is an opioid. Cocaine is not an opioid. It does cause a great deal of harm and a fair level of dependence. We have things such as tobacco and alcohol, which, quite legal, do cause physical harm and cause a certain level of dependence. And you may be wondering where cannabis is. Well, it doesn't cause as much physical harm, but it does occasionally cause dependence and addiction. So then you look down here, we've talked about anabolic steroids before. They cause harm, but they don't cause as much dependence. So this little graph is helpful to think of. But then we wonder about this current drug, fentanyl. Well, long answer short, it kills at a very low dose. And you can see that in this graphic. So why not take SSRI and MAOIs? Well, it says right there, do not take while abruptly stop taking medications. This actually causes something called serotonin syndrome. If you want to look that up, you can figure out why you have to watch what drug cocktail you're taking. So how do medicines affect neurotransmitters? They can bind to receptors to prevent the transmission of something. So if you bind to a receptor, then the neurotransmitter cannot bind, and that can actually stop the transmission. We didn't cover any drugs that did that. They can also bind to receptors to enhance the transmission, such as things like opioids. They can also reduce the degradation, such as monoamine oxidase inhibitors, and reduce reuptake, such as selective serotonin reuptake inhibitors. Thank you.